would be wonderful. Good afternoon, everyone. Welcome and thank you for joining us today for Season 2, Episode 1 of our Towards Zero webinar series. I'm Dr. Samantha Oakes, Director of Research Investment at the National Breast Cancer Foundation. Many of you are joining us today from the National Breast Cancer Foundation community. And for anyone who've missed our previous episodes, our webinar series is a way to keep you connected with our researchers, hearing firsthand about their projects, latest developments and exciting breakthroughs, all from the comfort of your home in this strange COVID environment. Today, we are going to be hearing from two of this year's National Breast Cancer Foundation funded researchers. Firstly, Associate Professor Belinda Parker, and secondly, Professor Paul James. They will be addressing the detection and management of early breast cancer. First up, we'll be hearing from Associate Professor Belinda Parker. Associate Professor Parker leads the Microenvironmental Crosstalk and Therapeutics Laboratory in the Cancer Immunology and Therapeutics programs at Peter MacCallum Cancer Centre. She is a Victorian Cancer Agency Mid-Career Fellow and previously an Australian Research Council Future Fellow. Professor Parker will be talking about her research project on predicting breast cancer spread. Belinda, I'm so excited to hear about your research. Please take it away.
Thank you very much, Sam. I hope you can see my screen. Very excited to be here to talk about some research that we were luckily funded from the NBCF on really trying to move towards precision or individualised therapy for women diagnosed with early breast cancer. So I just wanted to give an introduction to you all, just in case you don't know where breast cancer starts. So if we look at a cross-section through the breast here and look inside a breast duct, you can see that there's this really nice single layer of epithelial cells, and it's these cells that most cancers arise. In the earliest stages of breast cancer, the cancer cells start growing within this ductal system. And when they're inside the ducts, they're considered a pre-invasive stage called ductal carcinoma in situ. But when cancer cells start dividing out of these ducts, this is termed invasive ductal carcinoma. And it is at this stage that obviously there is more risk for invasion to other sites such as the lymph nodes, and therapy can be more aggressive than the early pre-invasive stage. Now, the reason we're really interested in early breast cancer is because mammogram diagnosis has really changed what we're detecting. There's been a huge increase in the number of early stage breast cancers, so ductal carcinoma in situ, detected. In fact, approximately 25% of cancers are now at this early stage that can't be felt by a lump in the breast often, but it can come up on a mammogram of calcifications. So the issue is that a lot of these women think they have full-blown breast cancer that is very scary. And we really need to know which of these women will go on to develop a secondary DCIS or another early breast cancer, or develop the invasive breast cancer that's gone outside of the ductal system. Now, the risk is about 30% of developing a second cancer. So that's really important to think about. We also need to think about which women are going to go on to develop the cancer spread to distant organs, because this is what causes cancer deaths. And the reason this is critical is because most won't. In fact, only about up to 4% will develop cancer that spreads to their bones or lungs or liver. So, you know, it becomes the question of who will actually benefit from therapy after a diagnosis of early breast cancer. And should we be sparing many women therapies that they don't actually need? And the reason this is critical is that because surgery, radiotherapy and hormone-based therapies can be very important for women with breast cancer, they're also associated with a number of side effects. Now, if we weigh up the benefit to risk ratio of these side effects, of course, if this meant that you had a decreased risk of cancer spreading, you would definitely take the therapy. But we need to know who won't have a benefit to spare women these side effects that they may not need or want. So I just wanted to show you what cancer looks like in a cartoon and also under a microscope. As I said, most cancers arise in these epithelial cells that you can see here. And there's been quite a lot of work on looking at the epithelial cells and saying, well, what's different in those cells that could really predict an invasive state later on? There hasn't been a lot of success looking at the epithelial cells alone. And in fact, what we've found in our lab is we can find much more information by looking at the cells around the cancers. And in this case, we're really interested in a cell type called the myoepithelial cells. And this is a single layer of cells that surround these epithelial cells in the breast. They usually function for contracting the breast ducts for milk production and release. And they act as a physical barrier between the epithelial cells or cancer cells and the surrounding breast. They function by attaching to each other, but also to the epithelial cells to keep them in place where they're supposed to be. And they also express factors that function to suppress the invasion of cancer cells. So, for example, suppressing the ability of cancer cells to express proteins that act as scissors to cut through the surrounding breast. Now, when we take a section through a patient's cancer or their normal tissue, we can look at a very thin slice under a microscope at about 100 times magnification. We can see these tumour cells or cancer cells here after staining them with different colours. And in the case of DCIS, it's sort of still within the structure, of the duct. But in invasive cancers, you sort of can't see this structure anymore. And one of the big differences is the absence of the myoepithelial cells. And you can see here in brown, this is a normal duct. We've stained the myoepithelial cells in brown. You can see this really nice single layer of myoepithelial cells. So what my lab has been really interesting is trying to find a way to see what are these myoepithelial cells actually doing and what is important in those myoepithelial cells that can really tell us whether there's been a change that could promote invasion in a patient. So if you have a look here, we've got cancer cells that are invading in a 3D situation. So we take a human breast cancer cell line, we can grow them in a dish in a sort of matrix or protein mix that's very similar to what's in the breast, and they grow, spread out and grow really invasively. Now, if we had myoepithelial cells that came from a normal breast tissue and put them in, we can see that actually keeps these cells in a very DCIS-like state or an early cancer state. They're not invading out. And if you look here, these red cells actually surround the cancer cells. So even though the cancer cells themselves are very invasive, these myoepithelial cells can keep them in check. Now, what we've done, we've used a screen to change what's in the myoepithelial cells to look for proteins that have a really important function. And you can see here that we've blocked one specific protein in these myoepithelial cells. And that was enough to allow the cancer cells to just keep invading despite those cells being present. So using this model, we've really identified cancer suppressor proteins, but they're not in the cancer cells themselves. They're in these surrounding myoepithelial cells, the barrier cells of the breast. And we think that these sort of proteins that we've identified could predict the risk of cancer recurrence and in the breast or elsewhere, and the benefit of therapy at a patient level. This is an example of one of the proteins we've found, and we've found many more that are part of this grant that we were funded for. We look at sections through the breast. This is a normal breast, and the brown is the myoepithelial protein that we identified. These are examples of ductal carcinoma in situ. You'll see that these ducts are filling with more cells. In some cases, there is the expression in brown of this protein we're interested in. In some cases, the expression's reduced, so you can see less brown. And in some cases, it's completely absent. And what we've found is that just by looking at the absence of this protein, we can actually predict patients that have a higher risk or a higher grade DCIS and those that also have a higher risk of getting invasion down the track. So I just wanted to talk about how we're putting this together for this grant. So what we're looking at is this funding has been given to myself, but also collaborators including Bruce Mann, who's a surgeon at Royal Women's Hospital, and Sandra O'Toole, who's a breast pathologist, And what we're looking at is combining our knowledge about these different myoepithelial proteins in these cells surrounding these breast ducts with the standard sort of variables that we look at when a patient is diagnosed. And this includes markers that are routinely looked at, such as the hormone status, the hormone receptor status, but also the immune cells that are coming in. These different colours here are just all different immune cells, and these are the cancer cells here in red. And what we've found is there's a really interesting relationship but with the immune cells that come in and recognise the cancers that is dependent on what's changed in these myoepithelial cells. So we want to put all this information together to build a microenvironment score. Now, this score is going to be used to predict the risk of early breast cancer coming back later. And we have large cohorts to do that through Bruce Mann at Royal Melbourne Hospital. We also have collaborators in the UK, and we also have collaborators that pharma companies that have large cohorts of patients to really look at our microenvironment score to see if we can tell a patient and the clinician, will this patient likely have spread of their cancer down the track or another cancer in their breast? Will they actually get benefit from therapies such as radiotherapy? Now, we'll be able to separate patients potentially into three sort of categories. There's one that don't need any other therapy. We could leave them alone. They're not likely to get a problematic cancer and they could be spared the side effects. Then there's the patients that will actually respond to radiotherapy and have a benefit, and other therapies such as endocrine therapies. But then there's another class of patients, and these are the ones that, despite these therapies, they'll still go on to get invasive cancers. And by looking at the changes in the myoepithelial cells, we're uncovering new therapeutic opportunities. We're knowing how this is happening, and we can target this as a new therapy going forward. So really, this all comes back to the 0 deaths from breast cancer. It's really fantastic that we're diagnosing more women with breast cancer at an earlier stage. What we need to do now is use this information to actually be able to treat them accordingly and then block the spread of those that will likely have problematic cancers down the track. So thank you very much, and I'm really thankful to the NBCF for this opportunity to do this project. Thank you.
Thank you so much, Associate Professor Parker. That was just a brilliant talk. I'm so thrilled to hear about your research and your incredible work and all you do to help NBCF reach our mission of 0 deaths from breast cancer. So we've had a few questions submitted by our NBCF community, and I'd like to just ask you a couple of those questions now. I think you covered some of this in your talk, but can you simply explain what these biomarkers are and how they will show if the cancer will spread?
Yeah, so it's really interesting. We looked at hundreds of different proteins in those myoepithelial cells, and what we're finding is that the ones that we think are critical for determining whether a patient is likely to have spread of the cancer, they have really important functions in binding those cells to the cancer cells. So the interaction between the two, keeping them in place and actually being able to work as a physical barrier to block them from moving around. So most of the proteins we've identified have some sort of function in keeping them in check, in tight binding, and preventing their ability just to go invade out. And we've actually looked in real time during a long process of, weeks, and we can see that these myoepithelial cells are really sucking them in, holding them tight and keeping them from spreading. So what we believe, and what we're working with pharma companies at the moment that have these sort of assays going where you can actually look at specific biomarkers in early breast cancer, and they're using them clinically, they're interested in a new test that would allow a patient to come in, get tested for these biomarkers, and that clinicians can then make a call on, will this patient need to receive radiotherapy? Could they be left alone or just with surgery? Or can we actually look at new therapies to actually restore what's being lost in these myoepithelial cells? So we're really excited about what we can do. We have some really excellent candidates already in the pipeline that are looking really promising.
It's really such important work. The second question submitted is, will your research help anyone diagnosed with breast cancer to know early on what direction their cancer will take? And what are the best treatment options using these biomarkers?
Yeah, I mean, one of my biggest aims is to spare women therapy. So at the moment, there's this it's not very consistent how many women are being treated with radiotherapy upon a diagnosis of early breast cancer. A lot of women will receive radiotherapy along with endocrine therapy, but it's really difficult to know who's really getting the therapy benefit from it. So the idea would be, and it's similar to one of the pharma companies we've partnered with, is how they sort of do it on their pipeline, is during your normal diagnosis where a biopsy is taken, the tests that we're using are exactly the same type of tests that are already being looked at under a microscope to look at the nature of your cancer. Do you have actually early breast cancer, or do you have an invasive breast cancer? And we can look at those same tissues for the proteins of interest with an antibody. So the idea is to put it into the normal pipeline and then to allow clinicians to use that score to decide whether a patient is really needing further intervention and will they get a benefit from that intervention. And, you know, there is definitely a pipeline that exists for this now. So you know, we're really excited to sort of bring that in, because at the moment there's no peace of mind for these women. They don't know, will my cancer come back later? Should I be receiving radiotherapy? You know, and potentially some side effects from that. And I'd probably be willing to take those side effects on board if I knew it was going to prevent invasive disease down the track. So that's the sort of the idea of where this would sit.
It's just amazing, you know, with breast cancer incidence increasing and we're detecting more cases, we can see how very important your work is. Thank you, Associate Professor Parker.
No problem, thanks.
So we'll move over now to Professor Paul James, also from the Peter MacCallum Cancer Centre. Associate Professor Paul James is a consultant clinical geneticist. He's the director of the Parkville Familial Cancer Centre at the Peter MacCallum Cancer Centre and Royal Melbourne Hospital, and a clinical associate professor in the Department of Medicine, Monash University, as well as the Sir Peter MacCallum Department of Oncology, Melbourne University. He's the group leader for the familial cancer research at the Peter MacCallum Cancer Centre. Wow, Paul, we're so excited to hear your talk.
Hi, Sam. Thanks for that. That's fantastic. I'll just see if I can share my screen. All right, does that come up okay?
All good, thank you, Paul. Right. So I just thought I'd introduce the title of your talk. You'll be talking about your research project, identifying women at risk of developing breast cancer using a new blood test.
Fantastic. Hello to everyone, especially everyone like me who's stuck at home and doing their work in their Ugg boots today. So as Sam said, I'm a clinical geneticist, so I'm particularly interested in the parts of breast cancer which are inherited or familial, and we think that that's somewhere around a third of all the cause of breast cancer is something that's inherited. But what I want to tell you about today is a story about how we got interested in something which is genetic but isn't inherited, and why we think that that might actually be quite an important thing to know about. So I'll get my thing, there we go. So today I'm going to briefly talk about what mosaic mutations are, and then tell you about why we think they might be an important risk associated with breast cancer, and how we could use that information to continue to identify the people at the highest risk in the population. So this is a busy slide, but it's really just making the point that a lot of the genetic changes in our bodies are not ones which are inherited, but they happen over the course of our lifetime, and they are found in different parts of our body. So we always think about genetics as being something that's handed down from your parents. And of course, anything that's a gene that's found in an egg or a sperm cell, so the germ cells, is passed down from your parents. And we can pick that up in your bloodstream by doing a genetic test. Normally, because we have 2 copies of every gene, we would expect any variation that we pick up in a blood test to have a frequency of about 50%. So one of the genes carries the variant, the other one doesn't. But we know that if a genetic mistake occurs at other times, it can have different impacts. So here you see at the top of this picture, we've got the idea of genetic changes which are in the germ cells being detectable at 50% in the blood. And at the very bottom, you have the idea of a genetic change that occurs somewhere in a cell in your body when you're an adult, and it's not a cell that's growing or expanding in any way, and it won't move anywhere. It'll just be in the cell that's particularly involved, and in which case we won't be able to find that just in a blood test. And in between, we have two special circumstances. One is where you have a genetic change that occurs at a very early stage in your own development, when you're just a little bundle of cells, in which case that genetic change may end up being found in lots of parts of your body, even if it isn't in the germline. So it can be scattered through your body in something that we call somatic mosaicism, meaning that we're all built up a bit like a mosaic picture of lots of cells, which are the tiles, and some of them can carry the mutations and others don't. And often when we do a blood test, we can see that as a genetic variant that's present at a low frequency. So there's a little bit of something. We're just picking up a few cells that contain this genetic variant. And there are some special cases, particularly where it affects the skin. And there's a little photo there of the famous sort of classic genetic condition where you get skin mosaicism and you have a whirling pattern of skin changes, which reflect the different cells that have different genetic makeup. There are also a couple of other special situations where a cell can get expanded or increase in frequency. And one of them is of course, the situation in cancer, where you get a genetic mutation in a cell that triggers that cell to start growing and dividing in a way that it shouldn't be. And that's the beginning of a cell starting to behave as a tumor. And then if it becomes aggressive, then it becomes a cancer. So those types of mutations, generally speaking, we can't pick up in the bloodstream because the mutation is confined to the cancer itself. And this is the case certainly in breast cancer, where if you want to see the mutations in the cancer, you actually have to do a biopsy to get the breast tissue. The other situation, which is a bit like that, is where the mutation occurs in the bone marrow, in which case we can see that mutation in the bloodstream. And that has a long-winded name called clonal hematopoiesis of indeterminate potential. And so understandably, we just call that CHIP. And CHIP means that we can pick up new mutations in the bloodstream at a certain rate that increases as we all get older. So now you're all experts on mosaicism. How does this relate to cancer risk? Well, we never really worried about somatic mosaicism in the past, mainly because we couldn't see it. So unless you had some obvious sign of it, we couldn't pick up mosaicism at all. And then around 10 years ago, the genetic testing technology changed, and we began to get a situation where we could see these low frequency genetic variations, things that are present in our body but not inherited. They're just in a small number of the cells. And you can see there up on the left-hand side is a picture of what that looks like when we do gene sequencing. You can see there's just a scattering of this variation, which is marked in red there. Most of the sequencing runs don't have it, but around 22% do. So that's not enough for it to be something that's inherited, but it's definitely there. When we do large-scale sequencing, we find that in fact there's quite a lot of these mosaic variants. And you can see there in the blue graph shows what we would expect, a nice sharp peak around 50%, but then it's got a sort of a little bulge on the left-hand side where you get all of these variants which have low frequency, and they're present, there's more of them present than we would expect. And then finally, in 2013, a group led by the Breast Cancer Association Consortium suggested that a variant that was present like this, that was mosaic in the blood, might be associated with breast cancer. And since then, there's been a lot of question about that result. People have asked whether or not it could just be an effect of the chemotherapy that these people, that the breast cancer patients would have had, or could it just be that they're measuring CHIP, which is a normal phenomenon? And so that hasn't really gone any further. Now, over the last few years, my team and I have been involved in this very large-scale case control study where we've been trying to find new genetic causes for breast cancer. And this study has involved 12,000 women over time and has allowed us to focus in on new genes which we think might be contributing to breast cancer. And recently we got to write up the results from this. But along the way, what we noticed is that amongst the huge number of genetic variants that we found, there was this group that were mosaic, that they had a low frequency, and that made up about 7% of all the genetic variants that we found. Except that in the people who had a history of breast cancer, it was more than 7%, whereas in the people who had no history of breast cancer, it was less than 7%. And although the difference wasn't huge, the actual number of variants was very large, and this turned out to be quite significant. So the way that we show that normally is a picture like this, where we're plotting where people sit if they have one of these mosaic variants. And the line down the middle of this graph is a risk of 1. So that's the same risk as everybody else. And you can see there that what we're trying to show is that if you have one of these mosaic variants, that your risk goes up. You have about a 20% increase in risk. But really importantly, if you have more than one of these mosaic variants, if you start to have a collection of them, then your risk gets quite high quite quickly. And so some of the people who have 3 or more mosaic variants in their circulation had a risk of breast cancer that was quite similar to some of the single genes, some of the genes that we've heard of, like PALB2 or BRCA2. And so it looked like it was quite a strong genetic signal for risk, particularly in that small group of people who had a number of variants. Overall, 16.5%, so around 1 person in 7 had one of these mosaic variants in their circulation. The big question, though, is what does all of that mean? It could just be coincidental, it could be artifactual, but in fact, we've done enough testing to now to know that that's not the case, that these are real mosaic variants. We've validated them, checked them out enough times to know that they're real. So it could be, as people have wondered, that a diagnosis of breast cancer leads to having more of these mosaic variants in your bloodstream. And so it could be a chemotherapy effect, and that's something that we need to consider. Or it could be that the mosaic variants themselves are contributing to breast cancer, that they're doing something that increases your risk of breast cancer. Or the third possibility is that having mosaic variants in your blood shows that you are susceptible to developing new mutations, and that it's a biomarker or an independent marker of your risk of breast cancer, even if it's not involved in the breast cancer itself. So how are we going to sort all that out? The first thing to answer is that question about, are we just measuring an effective chemotherapy? Because although that would be something, it wouldn't be all that useful in terms of working out who's at risk of breast cancer. So fortunately for us, we have access to this very special study called LifePool. And in fact, there's a good chance that some of the people listening in today are involved in LifePool, in which case it'd be good to know that you're making a contribution to the study, which is really very valuable. So LifePool involves around 50,000 women who are involved in breast cancer screening, who all joined the study when they were well and their screening was going well. But over time, as we followed them up, more than 1,500 women in that group have gone on to develop breast cancer. But what's very special for us is that this group includes people who gave us a DNA sample a long time before they developed breast cancer, and then have gone on to have that diagnosis later on. So we have this special resource of pre-diagnosis samples for individuals that go on to get breast cancer. And that's pretty rare because, of course, you never know in this world what's going to happen in the future. And the idea of being able to collect samples so that we could find out how breast cancer begins amongst people who go on to develop cancer is a really special thing. And so we were able to look at 74 people who were in that situation where they gave us a sample long time before they developed breast cancer. And we were able to find that not only did they have mosaic variants, they had even more mosaic variants than the whole group of individuals we looked at before. So the effects seem to be absolutely real and definitely present before people develop their breast cancer, meaning that it could potentially be a useful way of measuring breast cancer risk beforehand. So that rules out one possibility. The other thing I asked was whether or not the variants themselves are causing breast cancer. And we do know that in some cases, this is absolutely the case, that the variant that we're measuring in the bloodstream is actually present in breast tissue and is contributing to breast cancer. So here's this, although there's a lot of hardcore science on here, these are really 2 stories of individuals who were diagnosed with breast cancer at very young ages. On the top is a young woman who was diagnosed twice in her 30s, the first time at 31. In the bottom, a similar story, someone who's diagnosed twice at 30 and 35 years. Very striking stories, and made us think that they had a strong chance that they had a genetic risk. And then when we did the gene sequencing, we found that in both cases, there was a variation in the gene TP53, which is a major risk gene for breast cancer. But as shown there's the gene mutation, and as shown, it was at very low frequency. In both cases, a quarter or less of the genetic variants that were picked up showed the abnormality. So this appeared to be a mosaic variant. But importantly, when we went on to perform testing in other parts of the body, including in the breast cancer itself, we found that although, and it's shown down there on the right, in peripheral blood, in skin cells, and healthy parts from the breast tissue, lymph nodes, the variant was sometimes present at a very low frequency. When we looked in the tumor itself, it was present in a high frequency, and it was definitely part of what was driving that breast cancer. So these are examples of that situation I described of true somatic mosaicism, where the variant was present probably from a very early age during embryonic development, when someone was still a baby, was still a developing embryo, and then is present in different parts of the body and has contributed directly to the breast cancer. We've now gone on to show that that same scenario works for quite a proportion of these rare mosaic variants when they involve a breast cancer predisposition gene. So true somatic mosaicism, that is the variant causing breast cancer, is definitely a real thing. But at the end of the day, there's actually a large number of other variants where that isn't going to be true. In fact, we've looked at more than 8,000 mosaic variants in different genes in this group, and we can see that a lot of them are present in much more frequently in individuals with breast cancer than they are in the general population. So there's also the opposite is going to be true, where the genetic variant is not directly involved in the breast cancer, but it does provide a marker that somebody has difficulty with their DNA repair, and means that they have a higher risk of developing breast cancer in the future. And so that's where we're up to now, and that's where NBCF has come in to help us with this, moving this project forward. We're trying to perform what we think of as the definitive study in order to try and answer the question properly by carefully taking 200 individuals who've developed breast cancer from the life pool cohort, taking their pre-diagnosis blood samples and samples from other parts of the body, and then comparing that to post-diagnosis samples and their tumor DNA, along with other samples which provide our controls to compare to. And then we're going to go very carefully through all of their genes to work out how many mosaic mutations they had when they developed them, and which ones of those are providing us the best indication of future breast cancer risk, with the aim that at the end of it, we'll have a new biomarker that can be used as a blood test to determine people in the population who are at risk, even though they didn't inherit the genetic variants, that the genetic variants have come along at some point during their lifetime. So that's all I was going to tell you about, except to say thank you to all of the huge number of people that are involved in this kind of work, and particularly all of our patient group, who all of these studies are performed from very large patient groups, and it's only really the huge number of people who selflessly contribute to these studies that gives us what we call the power to make these kind of findings of very small, subtle effects in our bloodstream. So thank you very much, and thank you to the NBCF for continuing their support.
Thank you very much, Professor James, for that fabulous talk and for all the hard work that you're doing for NBCF to reach our mission of 0 deaths from breast cancer. So a couple of questions. If you're successful, when do you expect that this test would become available, and what exactly would it show to a patient?
Yeah, so that's always the key question. Where do I get the test and when? So of course, as you could see, we're really still at the stage of trying to answer that question about, is this a good marker? Does it really tell us enough about breast cancer risk to be included? And what we imagine is it would take its place amongst other things. So, you know, along with talking to someone about your family history, looking at things like mammographic density from mammograms, this would be a test that could be included in all of that, that would tell us about how good your intrinsic ability to maintain your DNA is. And the good thing would be that it could be taken, if we could do this test, for instance, when someone was starting on a program of breast screening, to tell us about what their level of risk was, to help us make a decision about how often they should be screened. So it certainly is something that we hope would be able to be implemented as part of that sort of spectrum of risk assessment. But of course, you know, we need to see some good solid results that take us down that path to show that, yes, it's very effective, and we've got some numbers where we feel confident to be able to tell people this is really your risk, so that you know, that it actually does its job when we use it.
Course, and it just shows the importance of research, because without the research we just won't know.
Yes, yeah, that's right. It's intriguing, but we can't get there.
Yeah. So if you are successful, and we expect that you will be, because you're fabulous, how accurate do you think that the blood tests would be?
Like all of these tests, it's a bit of similar to the way that screening works, is that it doesn't have to be really precisely accurate by itself. as long as it provides a good level of discrimination. So as long as it generally helps us to say, this group of people can feel reassured and don't need to screen as frequently, and that group of people, we have some concerns that their risk may be higher and we need to change their management. So it's not a case that we're never going to be at the point of being able to say, in the next year or two, you're definitely going to get breast cancer, or you can be 100% sure that you're not. It's always about shifting the risk around. And so the way that we think of this as working is, as I was saying, is being part of a portfolio of different approaches, and we would put it all together into what we call an integrated risk assessment, so that we take all of it into account and then provide a very personalized assessment of risk. And that's where we see this going, as being part of a suite of techniques that we've got for getting better and better at risk assessment, none of which by itself will do the job, but when you put them all together, they work.
That's brilliant. Thank you very much, Professor James, for that fabulous talk and your responses to the questions. So that leaves me to thank our wonderful speakers today. We're so excited about what you're going to achieve with the National Breast Cancer Foundation funded projects that you've been given. As members of the National Breast Cancer Foundation community, I'd like to thank you all, everyone that's here in the audience today, for your ongoing support of the National Breast Cancer Foundation and for joining us today. We hope you found this webinar interesting. And again, we'd like to thank Associate Professor Parker and Professor James for their insightful summaries of their projects. The webinar has been recorded today and will be emailed out to you later in the week. Thank you for your attendance today. Stay safe. See you later.
Thanks.
Thank you very much.